Welcome you all to this 3-day online national level workshop on DNA barcoding. Here myself, Adil Ahmed, Assistant Professor of Biotechnology from Islamia College. First and foremost, I thank Almighty for securing and strengthening my soul and blessing with successful journey of life on this earth. I would like to thank our beloved Principal Sir and our Honorable College Secretary Sir for providing me an opportunity to serve in one of the century-old renowned education institute in India. I feel much proud to be a part of this institution. Here I would like to thank the chief organizer of this workshop, Dr. A. Jafar Ali sir, for giving me an opportunity to deliver an online lecture on the topic of isolation of DNA. I would like to thank our department head Dr. A. Mubarak Ali sir and all other faculty members of my department. So, these are all the topics we are going to understand in short today. Well, you all know what is DNA. The life on nature earth, that is the all living creature, the all living one, from the smallest virus which is less than 200 nanometer in size, for example the COVID-19, which is now terrorizing and ruling the whole world, and to the largest animals on earth like whales, which is of 200 tons in weight, but is hiding somewhere inside the ocean, whose information are being coded and stored inside their cells in the form of genetic material, which is mostly DNA in all species or RNA in some viruses. Well, the DNA is said to be the blueprint of all the structural and functional characteristics of living organisms which are stored in it. Then we'll introduce you to the DNA isolation, why the DNA is isolated, that is the importance of DNA isolation, the working principle mechanism involved in it, and followed by the three basic methods which is using predominantly. Well, the DNA, as we said, it carries the information in a coded form, which we call them as a blueprint. So the structure of DNA, basically it is a double stranded helix. The structure of DNA is proposed by the Watson and Crick in the year of 1953 by means of X-ray crystallography technique. So they are basically the polymers of nucleotide. The three major components of a nucleotides are the nitrogenous bases, which are in the form of purine and pyrimidine. The purines are adenine and guanine, and thymidine, and the pyrimidines are thymine and cytosine. The pentose sugar is in the form of deoxyribose in DNA and ribose sugar in RNA. And it consists of phosphate group. So these double strand DNA are stabilized by hydrogen bonds and hydrophobic interactions. So basically the DNA is said to be negative in charge because of the phosphate group present in it. The occurrence of DNA is seen in the nucleus of a cell. And in some eukaryotes, we can get DNA from mitochondria and chloroplast in plants. There are some of major proteins where the DNA is folded and condensed into the form of nucleosome, then further condensed to form a structure of chromosome, which are being stored inside the nucleus. So some histone proteins. This is a general structure of double-stranded helix. So we can see all the molecular structures over here, the ribose sugar, the phosphate group, the nucleoside, like where the adenine hydrogen bonds with thymine, and the cytosine with triple bonds with guanine. And the length of the gaps and grooves. So we can see the minor groove over here, which is about 12 Armstrong, and the major groove is about 22 Armstrong, that is about like a 2.2 nanometer in size. A total helix turn we can see here, like it's about 3.4 nanometer in size, which is about like 10.5 nucleotides per 10 in approx. Basically, the central dogma of molecular biology we have to understand. There are three different processes are responsible for the inheritance of genetic information and for its conversion from one form to another. They are the DNA replication, the transcription, and translation. In replication, a double-stranded nucleic acid is duplicated to give identical copies. This process generally perpetuates the genetic information. Transcription. In short, we can say a DNA segment that constitute a gene is read and transcribed into a single strand sequence of RNA, like mRNA. The RNA which is transcribed moves from the nucleus into the cytoplasm for the process of translation, where the protein is synthesized. So in translation, the RNA sequence which is been released out is then translated into a sequence of amino acids linked to form a polypeptide chain, as they further formed into any of a structural and functional protein. During this translation process, the ribosomes reads the three bases codon at a time from the RNA and translates them into one amino acid. The number of amino acid, as we all know, linked to form a polypeptide chain. The number of polypeptide chain will go into a primary structure, then secondary, then tertiary. Then it will form a complete protein. Well, then we move on to introduction to DNA isolation. So DNA isolation basically says that the purification of DNA from samples. So some of the common source for isolation of DNA samples are normally our whole blood, even a hair sample, sperm, bones, nails, tissues, even dried tissues, blood stains, saliva, the buccal swab, epithelial cells, even from urine, some of the diagnostic paper cards used for sample collection, any bacteria, animal tissues, or plants. So, some of the factors are affecting the DNA isolation are mainly the pH, temperature, the sample preservation, and nucleases. Any kind of changes within these factors will affect the quality of the DNA to be isolated. Why do we need to isolate DNA? From the starting point of for numerous application which is ranging from the fundamental research to routine diagnostic process and therapeutic decision making in all the value of biological explorations, DNA plays a major role. For examples, to bring about genetic modification of plants, to study about the genetic modifications, to bring about transgenic plants, genetic engineering of animals, manufacturing a number of pharmaceutical products, for example, to develop vaccines, antibiotics, diagnosis of different medical conditions, any kind of genetic disorder, genetic fingerprinting technique, parenting analysis to find out the right offsprings. In forensic department, and also molecular markers. What are molecular marker? A short DNA sequence to mark or track a particular location on a particular chromosome. If we say in simple, there are two major type of markers in cells. They are nuclear markers and mitochondrial DNA markers. So some of the technique we've been used to isolate these nuclear markers in various by means of diagnostic aspects and in forms of various other applications. For example, RAPD, which we call them as a random amplified polymorphic DNA, AFLP, amplified fragment length polymorphism, and some sort of VNTRs, microsatellites, that is variable number of tandem repeats, which are about less than 60 nucleotide base pairs, and microsatellites, SNPs, which we call them called as single nucleotide polymorphisms, ESTs, expressed sequence taggings. So these are all type of molecular nuclear markers were seen. In mitochondrial DNA markers, there are some regions, control region, the D loop, which has a very potential applications in population genetics, The 16S rRNA and 18S rRNA sequence to study the phylogenetic analysis, ND1, ND2 parts to study interspecific variations, ATP synthase to study about population level differences, and CO1, CO2, that is cytochrome oxidase gene being using for DNA barcoding to identify the species, cytochrome B, which has also been used in species level studies. So these are the type of markers been using to study the genetic identity. Basically, in DNA isolation, there are three major steps involved. The first and foremost is lysis out the cell. We have to break open the cells. Then we have to remove the contaminants, that is proteins, RNAs and other macromolecules, which we can say precipitate, and then the recovery of the DNA. So the removal of contaminants we can say as purification, and the recovery of the DNA we can call it as precipitation form. This is a general flow diagram which shows the generalized steps involved in DNA isolation. Basically, from the sample we have got, it can be either any samples which I've mentioned previously. We have to go with this process. We have to go with mechanical lysis, with the help of chemical, with the help of enzymes, which to break open the cell wall, and also to break open the nuclear envelope, which to bring out the intracellular components along with the DNA of interest. Then we have to treat with some kind of proteinase, detergents, and about the very purification steps to remove out other contaminants. Then we have to concentrate the DNA. So again here we are using isoamyl alcohol and some salts to precipitate it. Then we have to preserve it with TBE, with tea buffer or within distilled water for further process and analysis. Some of the major instruments and the materials have been used in DNA isolations were tissue homogenizer, micropipettes, pH meter, tabletop centrifuge. Basically the cooling centrifuge is preferred, and water bath. The materials which we have to be aware of is sterile blades, forceps, tissue chopping materials, homogenizer tubes, centrifuge tubes, glasswares, tissue papers as well. So we'll go into the first step, cell lysis. We have to break open the cell by means of mechanical, chemical and enzymatic methods to release its cellular components. So mechanical by means of pestle and mortar, or by using tissue homogenizers. We have to grind, and then by treatment with various chemicals and enzymes to break open the cells. In cell lysis, we'll be using cell lysis buffer. These are all the major components which are there in cell lysis buffer. We'll see a simplest definition and use of each component, that is each chemicals. Sodium chloride in cell lysis buffer provides an osmotic shock, and also it regulates the pH of the sample, and also the osmolarity of the lysate. Then the tris HCl, tris or tris hydroxymethyl aminomethane is a common biological buffer which is being used throughout the DNA extraction process. During extraction from any number of sources, DNA is pH sensitive. During cell lysis, removal of unwanted cellular components and precipitation, tris is used to maintain a stable pH, which is around 7 to 8. Then EDTA. EDTA is ethylene diamine tetraacetic acid, a chelating agent which is widely used in molecular biology experiments to sequester divalent and trivalent metal ions such as calcium and magnesium. This ability prevents the DNA and RNA degradation, as the enzymes, metal dependent enzymes, which are mostly acting as a nucleases, become deactivated. Then SDS page. SDS, sodium dodecyl sulfate. It is a strong anionic detergent that can solubilize the protein and lipid that from the membranes. In addition to removing the membrane barriers, SDS helps to release the DNA from histones also and other proteins by denaturing them. So it disrupts the membrane as well as nuclear envelope. Proteinase K. It specifically cuts apart the histone proteins and subsequently dissolves the membranes. Proteinase K is an enzyme which specifically targets on histone proteins. Then removal of contaminants, that is a purification step followed by cell lysis, where the separation of DNA from proteins and cellular debris will be performed. Basically, we see two methods here: organic extraction, that is phenol chloroform, and salting out. In purification step of DNA isolation, the phenol, chloroform and isoamyl alcohol been regularly used. Mostly they will be like constantly they will be using in 25 is to 24 is to 1 formulation. So phenol, it dissociates protein from DNA, and in addition of phenol changes the solubility of proteins by forcing the hydrophobic non-polar residues, which usually hides on inside of proteins and away from the solvent to interact with the solvent. The chloroform. The chloroform denaturates the proteins as well as lipids. It has the efficiency it enhances the efficiency of phenol for denaturation of the protein. Here, the chloroform allows proper separation of the organic phase and aqueous phase, which keeps DNA protected into the aqueous phase. Chloroform denaturates the lipid as well. Isoamyl alcohol will be acting as an anti-foaming agent which protects the integrity of DNA. Then recovery of the DNA, which is about precipitation of DNA. Absolute alcohol, that is absolute ethanol, is layered on the top of concentrated solution of DNA. Where it brings the fibers of DNA which can be withdrawn with a glass rod in a form of thread-like structure. We can desalt the DNA with 70% of ethanol. So as said, the methods of DNA isolation, there are hundreds and thousands of methods are there. So we'll discuss the popular methods basically. Here, the three methods as we mentioned. The phenol chloroform method, the salting out one, and one with a commercial kit. The phenol chloroform method. As discussed, the lysis of cells using lysis buffer. Then the protein is precipitated using phenol chloroform isoamyl alcohol. Then precipitated with the ethanol. Finally, the DNA can be dissolved in TV buffer for further analysis. In salting out method, the cell lysis been carried out with the buffer. Then specifically the protein digestion is made by proteinase K enzyme. The protein precipitation is done by high salt concentration. The supernatant contains the DNA. The DNA is then precipitated by collecting the supernatant and adding the ethanol. The obtained precipitated DNA is resuspented in the desired buffer. So the kit method. One of the kit here explained is about Quagen kit. Here the method we use a spin column. The DNA is bound to the silica membranes of the spin column in the presence of high concentration of salts is being eluted out by washing out the contaminants, and the DNA is eluted from the silica membrane in water or low salt buffer. So with this I conclude my topic of DNA isolation in a simplified way.